Hello and welcome to this module 2 of this course entitled Basics of Immunology. In module 2 we'll be discussing about the antibody structure, types, antigen antibody interaction, and one specific interaction, that is ELISA. So these antibodies are the proteins which can protect our body from these foreign intruders or antigen. Whenever antigen comes in close contact with B cells, they get divided and they get cloned. So plasma cells are produced, and from these plasma cells antibodies are produced. So this is the antibody structure. We all know that antibody structure is Y-shaped structure, and it contains two heavy chain and two light chain. The two arms of the Y, they contain the hypervariable region, which is basically the antigen binding sites, and there are some domains like heavy variable domains and constant domains. The N-terminal part, or the terminal part of both the arms of this Y-shaped structure, is the variable domain, variable of heavy chain, variable of variable of light, and all the other domains are constant domains. So the first part, that is the arm part, that is known as Fab segment, that means fragment of antibody binding, and the last structure is known as FC region, or that is known as fragment of crystallization. So there are 5 different types of antibodies: IgA, IgD, IgE, IgG and IgM. Among them, IgA contain one secretory element, so that this IgA can be present in mucus and in tears, etc. Then we have IgD. IgE is basically associated with the histamine property, that means all the allergens and all, that is allergies and all are caused due to IgE. IgG, one specific very important immunoglobulin that is present, that can cross the placenta and can enter into our body, into the fetus body. Okay. So this is the antigen antibody interaction. Antigen antibody interaction happens in the Fab region, that is the hypervariable region, and the part of the antigen which is associated with the antibody is known as epitope, and the part of the antibody which is associated with antigen is called paratope. So in antigen antibody reaction, some properties of these antigen antibody reactions are, they are very specific in nature. Mainly there are non-covalent interactions are there, and two terms are very important. One is affinity, another one is avidity. So affinity means how strongly an antigen can bind with the antibody, and avidity means how the antigen antibody complex are bind together to each other. So it depends on antibody's affinity, it depends on the structural configuration of antigen and antibody, and how epitopes are interacting with the paratopes. Also there is one term that is known as cross reactivity, means one antigen can bind with different types of antibodies, and one antibody can also bind with different types of antigen, but there is one single catch, that is there should be structural integrity or structural complementation. So these are the types of antigen antibody interaction. So antigen antibody interaction may be of precipitation, may be of agglutination, may be complement fixation, it may be immunofluorescence or ELISA. ELISA means enzyme linked immunosorbent assay. So there are 4 different types of ELISA, that is direct ELISA, where we use one antigen, we give that complementary antibody, and that antibody should be attached with one enzyme, and that enzyme, when we add one substrate for that enzyme, there will be formation of some colored product, that is called direct ELISA. In indirect ELISA, we use 2 antibodies. For the same process, we use 2 antibodies. First one is known as primary antibody, and second one is known as secondary antibody. In case of sandwich ELISA, first antibody is there, then with that antibody some antigen will get bind. With that antigen, another second antibody will get bind, and with that second antibody, third antibody will get bind. So in the direct ELISA we have one antibody, in indirect ELISA we have 2 antibodies, in sandwich ELISA we have 3 antibodies, and again we'll add some substrate, and if the antigen antibody reaction has occurred, then there will be some colored product will be formed. This is sandwich ELISA. The fourth one is competitive ELISA, that means we'll use one antibody and 2 different types of antigen, and we'll see with which antigen that antibody gets bind at higher affinity and which one is there with a lower affinity binding. So that is the competitive ELISA. So this was the module 2. Thank you.